something slightly different. Designing matter and mimicking life. Zorana.
Thank you, Simon. So thank you, first of all, thank you, Adil and Simon, for inviting me to be a part of this workshop and conference. This is the first longer one I attended since COVID days, and it's sort of nice to see. Actually, I see how much I miss things like this. It's very, very different than on Zoom, even though it's more convenient. This is something very different. So I've been enjoying immensely. And so before I start, I'm going to do something I've never ever done in my entire life, and this is to do some shameless advertising because I want to. Why does it not work? Because I want to apologize for missing out on all the social events in the evening with you. So I've been avoiding everyone because there were some last minute things we had to do for the book that I've been writing with my colleagues, Carlos and Chancellor Dalit. Free textbook for undergraduate and graduate students, has lots of exercises. There's an accompanying website with lots of things. My co-author and publisher told me that I should do this, I should advertise. So I'm doing this for the first time in my entire life. I've never advertised anything like this before. So, but this is the reason why I've been not attending any of the dinners, because something came up and we have to finish it super quickly before things go into publish.
There's a fee for advertising, and you know what it is? It's dinners.
Is the dinner? That's a good idea.
Now you feel completely innocent.
Okay. Okay. So now back to actual topic. So this is going to be slightly different than what we've heard so far. It touches a little bit upon the talks that Reidun and Greg did last week, and also Michael and who left. But because many of you don't know me and don't know what I've been doing or what my research is, I'm going to first introduce it just a little bit to give you an idea about how we are thinking about our packing systems. Okay, so we are inspired by life, okay, and in what we do. So, and life as we know it exists in multitude of forms and shapes. But when boiled down to the very essentials, it's basically these most basic elements: hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus. And if we look at things like nucleic acids, RNA, and DNA, they are made up out of all these elements. Then if you look at some amino acids and proteins, they contain elements up to sulfur, some others up to nitrogen, carbohydrates and lipids up to oxygen, and of course, hydrocarbons are collections of carbon and hydrogen. And now the point is that all of these, very few of these elements actually make up all these living things. So
you want iodine.
Sorry?
You want iodine. It's pretty heavy.
Yeah, well, yeah. And there are some heavy stuff here. I agree with you, but very roughly, it's basically these things. And they make up for all these different shapes and forms, like this tree here, echidna, this is leafy sea dragon, and dragonfly. So life forms very diverse and very broad. And we sort of, they evolved right in time, but they are sort of dominated by accident and things that were good at some time in the evolution. And so many scientists, since forever, have been studying nature and trying to understand what are the laws and rules of things that we see behind what we see in nature. So what we do is slightly different in a sense that we are not trying to limit ourselves to what we see in nature, but we are driven by something that Christopher Langton said some years back, and this is to have a theory of action. It is necessary to understand what is really possible to do. So we are inspired by nature, but not limited by its sets of solutions. We are not limited by the number of building blocks is available. So we don't live just in the chemistry of life, but in any artificial chemistry. We try to explore all the possibilities. And so what we are doing, we are trying to find what are the necessary ingredients that we need to achieve certain lifelike property in physically realizable systems. And we try to understand how to control different levels of organization. And the goal of all this is, of course, to find efficient and robust design rules at different scales. So basically, we start from something that's very abstract without any constraints. We have artificial atoms, artificial chemistries that have no constraints, and then we introduce some physical constraints and then try to find minimal ingredients that lead us to some emergent behavior. Okay, so that's very roughly what has been driving my research for the past years. And so this is again just an advertisement of what we do before I move to programmable folding. And this is more to stimulate if you want to discuss later on. So we work on designing artificial catalysts made out of colloids, then trying to understand what makes an artificial chemistry good chemistry. We're trying to understanding how we can self-replicate structures that have a bulk or don't. Then on programmable folding, which I will tell you a bit more about. Yes?
I have a simple question. How did you find life?
We don't. I'm not going in there. Absolutely not. So, I mean, there is no way I'm going to try to attempt to do that. That's why I'm saying lifelike properties. We are mimicking lifelike properties. Okay. That would be I can be more specific, but we are mimicking lifelike properties. I mean, in lifelike, we know it, you know it when you see it. I mean, when you say replication, this is something that you know is something that's proper to life. So when I say, so in that sense, we are mimicking life.
There's a definition of biology that I say if it can self-replicate without the help of something else alive. So virus often uses growth factors to replicate, so what we class as not alive, but something that actually can self-replicate is often classed.
Yeah.
This is the best thing I can come to.
Yeah, so this is how I see, but I'm being very partial. I'm not trying to define life, I'm just saying mimicking lifelike properties.
But they are self-replicating.
These are self-replicating.
They do self-replicate.
They do self-replicate, but I wouldn't classify them as life. Yeah, and I also don't think this is I don't think this is also like a jump thing. I think this is a continuous thing. I think it's very hard. I don't think it's. I think it's continuous. So they do replicate.
Can I help? There are many properties of life. We know quite a few of them. You're looking at them perhaps one at a time or several at a time. But maybe there's some that you don't really know, and those are the ones that
Sure.
until you replicate, mimic those, you don't know whether you mimic all of life.
Well, but for sure. But it's also the thing that in the system that we do, these things, some things emerge even without us actually putting things in. So this is what is the exciting part. Okay, so. But we also are interested in designing matter, and this is so we are interested very much in quasi-crystals and how we can assemble them from what we call foldamers, and I will talk about them later, and then also use the same foldamers to assemble shells. And so I put these because these were last week in lectures by Michael and Andre. Okay. So that's just an advertisement. So if you have any, if you would like to discuss about any of the topics, I'm very happy to do that. Okay, so living matter. So this is, if you haven't seen this before, but I'm sure most of you have, these are David Goodsell's molecular art images, which are just fascinating. It doesn't matter if I've seen them thousands of times, whenever I see them, I'm always at awe. And it's trying to depict basically here just how molecules are crowded in living cells. And here are just depictions of individual molecules. So what you see here is sort of a crowded environment, right? Where many things are happening, but most importantly, things are being created, things are being self-assembled all the time, very efficiently and very robustly. Although in environments like this. So there is this process of self-assembly, spontaneous self-assembly of some building blocks into complex structure is very important for living systems. And so this is the property that we are going to be focusing on today. So self-assembly, everyone knows, it's ability to spontaneously assemble and make complex structures. from smaller, simpler parts with high fidelity and high yield. So what I'm depicting here are just amino acids that together with this piece of RNA can make the smaller subunits of the ribosome. So the red pieces that you see here of the RNA are the specific places where some of these amino acids in forms of protein complexes come in and bind. So there are some specific interactions between building blocks, RNA, that lead to making this very interesting machine. So how can we try to mimic something like this in artificial systems? Well, we want to understand how we can assemble things robustly and efficiently. And for that, we move to toy model systems, which are what Adil and others were mentioning also last week, are sphere packings, both in 2D and 3D. So this is the model system that we were working with. These are packings of small of spheres, of identical spheres, which mathematicians have been enumerating for many years, and we sort of know what they are. Depending on what number of spheres you take, what is depicted here, are all the different structures that you can make with them. So here at 18, there are 13 different structures. Here in 3 dimensions, in 2 dimensions, there are only 9 at 8, but of course they grow exponentially as you grow in size. Now, the nice thing about this is, as I said, is that they are really all enumerated up to certain size. So whatever questions that we are asking about what is possible, what can be done, we can ask on the whole set of possibilities and then get the answers out. Okay, so let's say that we want to build so if I have identical particles and I take 9, I can get any of these, I don't know, 52 structures that are drawn here. But what if I want to get one specific one? So how do I do that? Now, looking into biology, we know that the way that it makes these very complex objects is by putting specific interactions. So by having specific places where things can bind. So in our toy system, what does it mean? Well, it means that we have to take different particles. They cannot be all identical, but we have to somehow give them identity. And if we give them identity and give some interaction rules, which is basically depicted here. This is an interaction matrix that tells you which atom type is interacting with which and in what way. Then what you can find solutions like this that allow you to build any of these specific structures uniquely. So if I take 8 particles following these interaction rules, I will get this structure and none of the others made out of 8 particles. This is just an example in 2D as well. So we can make different arbitraries, we can assemble different arbitrary structures. I mean, this is just, these are sketches, but people have been studying this in theory, in simulations and experiments. It works. If you take many different colors, you have a design that are designed to make one specific structure. If you do an experiment or simulation, you will actually get that structure. But to have this reliable assembly really at high yield requires that you have many different building blocks. So here is something with 15. You need really 15 different building blocks to make this structure. Ribosome now has like 70, but that's a ribosome, right? This is just a small sphere package. So something we can make it, but it's really expensive, right? There are many ingredients that you need. And we know that, of course, nature doesn't do anything with that many building blocks. It does with much fewer. It just uses 20 types of building blocks, but it uses polymers and sequences of these amino acids that then fold into desired structure. So there's a way to encode information about different packings in the sequence of building blocks of certain colors. And so this is what we decided to try to do. So we know how to assemble things from gas, from many different building blocks, but we wanted to try to assemble things from chains and try to see how well we can do with the fewest possible building blocks. Okay, so that's the task. So what are the tools that we have, what are the ingredients that we have available. So again, with these sphere packings working through the 3D, you have basically everything. You can get the free energy. You can get the free energy minima. You can do the free energy landscape. You can find like what I'm showing you here. This is just for small systems, but we can do it for up to any size that we have. So it's a free energy, and these are different structures at every level. With red, I'm just marking structures that have the most bonds within the group. So here at 8, there are 9 different structures. There is one structure that has more bonds than everyone else. Of course, it's also lower in the free energy landscape. Here at 6, everyone has the same number of bonds, so they are all colored with red.
Could you just specify whether your particles are hard or soft, and what kind of interactions they have to define this?
So for the mathematical part, which is the database that we are using, these are just considered to be just hard particles, right? That you're packing hard particles. But in simulations and experiments that I will show, these are going to be sticky spheres that interact with a very, very short interaction range. So like a 1–2% of particle diameter. So very, very, very sticky. A very short range sticky sphere. But for these enumerations that we are doing, and whatever we are doing on these packings, they're all just come out from considering hard spheres.
Will it be almost the same if they are soft, or is this a new problem?
I would say that's a different, slightly different.
So if these are hard, what's F then?
Can you repeat?
What's the free energy then if they are hard? I mean, is it only entropy? Or is it actually... Yeah,
exactly. So you can calculate the rotational entropy, the vibrational entropy, and the rotational entropy of all these things, and separately. You are making an assumption of they're interacting via some harmonic potential, but very, very, very short, very short. Is that okay? Okay. We can go into details later on. The point here is just that we have this information. So what other information we have? We have something that we call folding trees, or folding trees. So basically, starting from a chain of particles, we can find what are all possible pathways that will lead to them folding into a compact structure. So in this case, we're looking at a 2-dimensional system. So for example, if we are 7 particles, there are 4 distinct geometries that can be made with identical particles. And so this is a folding tree that shows that if I start from a chain, so this here, there, here identical, and then I follow this black path that I marked, I will end up here, which is exactly, I think it was this geometry here that you see, this ladder-like structure. Now you can see that I'm showing you here 4 structures, and there are actually many more dots here at the end of the tree, which are the compact structure. And this is because, and this is something that was hidden a little bit in Raiden's talk, we have to look at all possible ways in which we can fold actually these chains. So Hamiltonian paths are very important to enumerate as well. And they grow even faster than the number of structures as a function of time. But anyway, so these are the information that we have. Okay, so now we actually want to assemble something reliably. So we need to put some specific interactions that we said already. And we're just going to start with 2 and nothing more than that to see how well we can do with 2. So 2 types of building blocks, which means we have 3 interactions. So these are blue and yellow, blue likes blue, yellow likes yellow, and then blue likes yellow. And for the things that I'm going to show you, we are going to be mostly focusing on these alternating AB sequences, mostly because many of these things were motivated by experiments done in Jasna Brujic's lab, which I will show later on. But all the calculations we've done is actually much more broad, both in sequence and composition space. Okay, so these are the polymers, and then we have one more control now. It is not really nature-like, but this is what we have in experiments. So we have something that's called temperature protocol, which means that we can control when certain interactions are on and when they are off. And so this is going to be, this together with the sequence is going to be our main tool to control which pathways are selected from these folding trees. So to be more precise, if we start from this AB chain and we design interactions like that, if we start lowering temperature, we turn on first the yellow likes yellow interaction. From all of this huge folding tree, the only paths that are possible are these yellow guys that uniquely end up in this structure that you see here. If I now turn on blue likes yellow, I can make a few more, I can make one more step and go here. And then if I turn on blue likes blue, I end up in a very specific ground state. So it's not the structure that is at the bottom of the tree. So it's not the structure with the most bonds. It's really the protocol and sequence selected something that's not the bottom of the tree. It's not the 300.
Yes. So it's binary in the sense that you say it's either this probability one interacting or not interacting at all. But if you have certain probabilities, so the yellow it likes with a certain probability, then the less so it likes blue yellow, but it still likes it. So then the relative probability should play a role. And
yeah. So again, this is also just enumeration algorithm design. I will show you simulations and experiments how this in reality really looks like, and it doesn't matter. Okay. That's the whatever variation, small variation that you're talking about, just doesn't. It doesn't matter. It's very robust.
Yeah, and you call this temperature protocol. So in your experiments, this is really a function of temperature.
Yeah, exactly. So that's why we have it. It comes from experiments purely.
Yeah. And then first you have just the red one interacting, and then you allow for the blue one. So it's like you step by step. You first allow the red ones to interact, and then you allow the blue one to interact. Or do they interact at the same time?
Yeah. So let me. Okay. So this is how it works. So I turn on yellow. It's active at some temperature. I lower the temperature and turn on the next one, and even more. So it's downhill folding, and I have a protocol that just lowers the temperature. It takes some time to do this, and basically what it does, it selects a unique geometry which we call a foldomer. Okay. Again, this is just coming from enumerations. And so what we can do really, we can try to find all the unique assemblies that we can find and get as a function of system size for alternate sequence in random sequence with 3 colors and so on. So with the black, I'm showing like here on top here, what are all the possible number of geometries you can have at different sizes. And what you can see is that, okay, so with 2 colors, you can do to some extent, you can design certain of these structures, not all, but if you already move to 3 colors, like more than half of what is possible up to 13, you can actually make. And so this is really striking because, I mean, if you just take 2 building blocks in a gas, you're going to get a crystal, right? If you take a smaller number of particles, you're still going to get many different objects, right? But here, by encoding things in a sequence and protocol, you actually get very unique structures in a controlled place. So not a lot of ingredients to get something.
What is that white pink?
Here? This one? Yeah, it's because at the time when we were doing this, these were not done. So this was just an estimate. This was this is a combinatorial nightmare. So well, we haven't updated it, but it's a combinatorial nightmare.
And then if you have 3 ABC, you could reach to the maximum.
So you
you
stop at 11 for everything else. It's plateauing. If you take 4, of course, it recovers. But so but anyways, we were focusing at 2 because of course beautiful experiments that are done in Jasna's lab, and some of the motivations of how we design interactions really come from this experiment. So what they do in experiments is they take colloidal, well, these are emulsion droplets. They are around 2 microns. These ones are 2 micron size, but they often work with 1 micron size particles. They have several types of DNA on these emulsions. And now the beautiful thing about emulsions is that because they're emulsions, these DNAs are actually mobile on the surface in the lipid layer, so they can move around. So what they can do with this system very easily is they can actually control valence, although it's not necessary in this particular experiment. This is also what can be done.
So I can ask another question.
Yeah.
How long can the AE sequence be? Because if it gets longer and longer, there are different interactions even yellow can do, right?
I show, no, so they can make them 20, 25, it just,
So there's
they can make them quite long. So things that we were experimenting with, I think also 13. I will show you in so no, don't apologize. I'm glad there are questions. Okay. So they have what they call a backbone interaction, which is independent of everything. This is how they make them, and this never breaks. So this is how they get chains. And then they have these other DNAs on what we call secondary interactions. And so these are the experimental details, and this is how a snapshot of a part of a view field of view looks like. So you can see. You can see some very long ones here. And so there are chains of different lengths. This is from a few experiment statistics. This is counted as a function of system size. This is a log linear scale. It's exponential. And I don't know how much you see the numbers, but you can see that around the system size that we will be mostly looking in experiments, which is 6, 7, 8, 9. They can, in a typical experiments, they have 100 different chains, which means you can really do statistics on this. Okay, so how does the experiment look like? We start at high temperature. The backbone is stable at high temperature. We then lower well, not me, but Angus and Jasper Live, but I don't do any of this. We lower the temperature, which then allows for the blue-blue interaction to become active. I will show you a movie later on of how this really looks like. Things move around, make bonds. At some point, we lower the temperature even more. Blue-yellow comes on. So blue-yellow now start to interact with, they're not already bound by a backbone. And then at the last step, we turn yellow-yellow, and we end up with a compact structure. And so now, we can do various things. So one of the things that we did is we give them our folding trees, and then they see what they can actually see in experiments. And so this is what I'm showing you here. So in the background, as you can see, we started with something that comes out only from our enumerations, and then in wherever you see a picture is really a state along the folding process that they observe in experiments. And they actually also have statistics of states. So they know which one are the most popular one, which are not. So we have kinetic information from these experiments.
Sorry, but you show us now for 10 minutes, you keep the temperature and then you lower the temperature. Does this time has an effect, like you did it for 20 minutes and then reduce the temperature?
Yeah, so I think typically these things are very fast. So the interaction, they bind very, very quickly. I think after 5, 6, they already see everything locked in. So there is a little bit of binding and unbinding, but in the end they like to make bonds, right? So once they have 3 partners, they just lock. Although one partner locally they did binding and unbinding, but then as soon as you have 2 or 3 that come together, they sort of lock. And it's much harder to break. And typically they leave them for 10 minutes, they can do 20. I mean, it doesn't deteriorate, it doesn't change dramatically the outcome. Okay, so they have statistics on how things are actually folding. So this was a sort of a populated tree. You can also have something that's even more reduced compared to the original tree. So it's again 7 particles. So here we started with blue, and then end up in this geometry. If we start with yellow, we end up in something that's floppy, and then depending on which interaction we turn next, we can sort of end up in different geometries. Okay, so that's the summary, at least, of experimental structures that were observed when we did this. And so on the top are yields that are measured from experiments, and below are the ones that we measured in simulations. And so usually in these standard business, unless you're making a crystal, if you're making some arbitrary structure, 100% yield with 2 ingredients, it's impossible. You need to have many, right? But clearly sequence does quite a lot, and you can reach 100% for many. And for the rest, it's actually not that bad. For the ones that are lower, we understand why, but that's a different story. And so what we also should figure out is that there are different holding modes, so different ways of how these things are made. So it's something that we call core collapse. This is a sort of a standard thing that people know in polymer. So you just have a core of one color that is surrounded by a shell of another. Or we have something that we call geometric frustration, where you have these checkerboard patterns where the first step is the interaction between blue and yellow, so not within the same color, but between the other color. And then it's enough to just make one bond between 2 particles, and then everything else locks in immediately because it cannot go anywhere. And so this is what's depicted here. So if these 2 come together, everything else is locked, even though they're not interacting at that point in time because the temperature is not right, but it's completely locked. Yes?
It's slightly just confused about the chemistry of DNA interactions, but you have something like yellow interacting. They're not complementary DNA base pairs, so how do they attach to each other? I would have thought it would be something like blue-yellow would be the only ones that are attached to each other.
No.
I'm just like confused.
Yeah, no, that's okay. So this is the backbone. So this is the rest of the interactions. So this is an example, right? Because depending on the protocol, whether we want first yellow to be on top or blue or blue yellow, the batches are different. Okay, so then maybe this is where the confusion comes in. So this is an example where the blue interaction has the highest melting temperature, and so you can coat the blue and blue particles with palindromes, with palindromic sequences.
Oh, I see. Very nice.
And then at 40, they lock in. Then you have the blue-yellow, which is just a 6-base.
Yeah. Thank you very much.
And then the yellow.
Very nice.
Yeah, they can do amazing things there.
There's something else interesting.
Yes.
Sorry to interrupt you. I thought it was a really cool talk. So the n equals 9, n equals 10. If you look at the geometries, there is only one yellow different. Otherwise, from the particle of the sphere, it looks the same. But then I tried to work out the half in the corner, so the order in which they are. So maybe it sits in a different order. But it's interesting that one on 9 is 100%, the other one is 38%. There's just one yellow away, right? So I tried to figure out why that is, because from the protein interactions, just one extra yellow going in, right?
Yeah. Okay. So the thing is that apart from just choosing interactions, this one likes first, then the other one, which is really boils down to how well we can design DNAs. There was no optimization of any kind involved. So what I can show you, maybe that's actually I should do.
I guess to rephrase what I'm saying is the kinetics of how it gets there, apart, not just the temperature, because otherwise it's just the bond counting, but it's not. It's actually the order in which these bonds are initially counted.
Yeah, but the thing is that okay, so this is the point. We get just a tiny bit of an optimization in terms of when you turn on the temperature. This crown thing goes up to 80%. So it's really so we were here, and if we were just doing it a little bit, because again, it was no any kind of optimization was not intended. It was just okay. We start from what we have, and that's what we look at. But the thing is that the other one was fine with the selection that we had. The crown, which is again just another one, it's just a tiny bit of a weaker interaction, you immediately get a very high yield.
Yeah.
And then what I didn't say is that you don't mess up the yield of the other one at the time. It stays good as well. Okay. Okay. So the folding modes, and then of course the best thing is to actually show movies, right? And this is simulation that we can do, but this is the real deal. So these are the experiments. And so what you can see here, they bind and unbind a little bit in the process of minutes. Maybe this is sped up, of course. I don't know by how many times, but in principle, it happens quite fast that you get. Okay, and so these were the experiments, but as I said, in our algorithms and simulations, we can go beyond the alternate sequence. We can go, we can look into the sequence space. And then what is nice is that we can find foldamers, which if we fold them and then collect many of them together, start forming self-limiting structures. And this is very nice. So these guys fold with pretty high yield. And then if you just put a lot of them together and just continue turning on interactions, they make tubes. These ones make what we call space invaders. If you don't know what space invaders are, you're very young. If you know, I'm not going to say anything else. But the point is that you can actually get self-limiting structures with not a lot of ingredients, right? That's the whole point.
Are these structures also like the ones on the left? Are they also assembled from a chain?
Exactly, absolutely. So I'm not showing it here, but we start from a chain. We turn on interactions that make it into this bed structure, and then we turn on another interaction that makes it.
It's not an AB, something else.
No. So this is an AB chain, but. It's still okay. So it's still. So no, sorry. This is not an AB chain. I'm sorry. So this, yeah, it has yeah. That's why. It has blocks of colors. Yeah, exactly. It's not sorry.
Then in this simulation, you see that the two of them on top of each other, they all like kind of sliding,
yeah,
bind and unbind, bind next to them.
Exactly.
And how efficient it makes a tube then now. You see, it's always one when it wiggles around with the bottom, you see.
Yeah. So again, there was no optimization that was done here at all to try to really find the exact ingredients that are the best for this system. You are absolutely right. It goes back and forth because you just made one contact. So it has a wiggle room, but as soon as it locks, it makes two. It doesn't.
But even when it makes it, you see, still it binds, and then again.
Yeah,
it
could be because we do keep this temperature for some. Okay, so you're right. It needs to lock. It needs to make more bonds to completely lock.
How do you select the number of elements you have in the chain? Is AB affinity could do chains of any length? So how do you produce the initial polymers which are linear of a given length?
So I'm not sure.
If you say I assemble chains where you have, for instance, AB,
yeah.
Out of the monomers, you could have just one more monomer that links up to the rest, and you might have rings, for instance.
Yeah, okay. So in experiments, they really, when they make them, because they have a system with some background magnetic field, the way that they align these things, they really end up with a batch of things of different lengths. This is the histogram that I showed before, some exponential distribution of chain lengths. So when they initiate a protocol, it's very painful job. They follow everything to try to understand what is happening. And then from the ones that are of the same length, they analyze what happened. So you can end up with many different lengths. This is true, but what the data that I showed, the analysis that I showed, is just focusing on single lengths at the time.
I pick out of the assembly those who are interesting. But if you do you supercollidal assemblies, you need lots of similar ones coming together.
Yeah, well, not necessarily.
No?
Okay, so here I show this is still in its baby steps because this is more complicated. As I think it's clear also from what you're saying. So this is an example where they did not do a pre-selection of sizes. They just have a batch, and they know the way that they made it. They know what is the distribution of sizes. And given the protocol, we know what we can expect to see in terms of what fold numbers we get. And then this is just an example. Again, there is no control. This is where we started with turning on sequence of interactions, this ladder form, but another tetramer formed, and they come together and make a longer chain because of that. What they're working on experiments on right now is our systems with microfluidic devices where they have sequence control and length control. So they can end up with whatever mixture they want in a given batch. Here there was no control of any kind, but in principle they're doing. I can maybe show. Sorry, just do this. This is one of the beautiful pictures. So here there is no sequence control, but they can make whatever they want. And now in their system, they can actually even select what sequences and how many in which batch they end up. So if you know what ingredients you need, they are developing a system where they can decide what ends up in a batch. And then you were asking a question about making a ring. This is entropically really unfavorable. Okay, that is fine. You do see them. You can see them, but very, very, very rare. Yeah. So I mean, on this log scale, the orange one you barely see. It's because it's a two-dimensional system, and this AB it's just harmonical. Like they want to just locally bind. This costs a lot, so that you don't see that. Yes.
Your simulation are all 2D,
so we do 2D, 3D, 2D to 3D. Just with simulations, it's easy, but they're just simulations. In experiments, they actually can do all of these things now. They can also do 2D to 3D, directly 3D. Yeah.
The experiments point given like
yeah.
3 and then 1 comes on top of it.
So they
always look at 2D and they don't look at it doesn't look like a blue one is on top of a 3 blue.
Yeah. So they do beautiful density matching. And they can do it on the third decimal place. It's amazing. So all these particles float on the same height in their sample.
Make sure that they don't fall.
They don't. It's just that it's so beautiful. Like it's literally third decimal place, but it's just gorgeous. And they just float there for the time of the experiment. If we wait for 3 hours, sure you'll start seeing up something. You can see them a little bit, right? It's not entirely, but they perfected it now. It's just like it's gorgeous what they can do. So yeah, so these ones, but then it's easy. You know, if you want to get to the third dimension, just flip the sample, let the gravity do stuff for you, and then they can explode.
Yeah, it makes it a bit hard to be on, but that's some more possibilities for happening.
Oh, it depends on what you do. Okay, and so what I've told you so far is the work that we've sort of published and we understand a little bit. And everything that is coming now, which is also related to some of your questions, you take with a grain of salt because you're still exploring. You're not 100% sure. We don't understand everything, but there are interesting things here. And they're related to patterns and geometry, and I think this will be hopefully really interesting to more of you. Okay, so there's something that we call pattern designability. There are some geometries that are more easily accessible than others. What I mean by more easily accessible, if I fix the system size and I look at all possible sequences, both in sequence space and composition space, And I look at which ones, given the protocols I have available in experiments, actually lead to something compact at the end. I end up with a graph like this. So if I focus here, I don't know, 12, there are 155 different geometries here. There is one geometry. So there are maybe 10 geometries that you end up making out of these 155, but there is one geometry that uses up most of the coding sequences. And then the rest, maybe one sequence per geometry or two, but they're definitely preferred geometry. So this is actually a preferred fold, but sorry, why is it not? Yeah, this is a preferred fold. Sorry, this was supposed to be the slide. But then this is a differently cumulated the same data, which is then preferred geometry. So you really have, there are some geometries that are more designable than others. And so we've been trying to understand which are more designable and why are they more designable. And at least for two-dimensional systems, up to some size that we looked at. So I come back to this free energy landscape picture of these different system sizes. And what I've circled in red are the structures that have this dominant bar height in the plot. So the ones that end up with being the most easy to code for in sequence and composition space. And they all end up being thermodynamic minima, which is sort of, is it intuitive or not? I'm not quite sure, because before, when we had this just downhill folding, which we are doing also here now, we were not ending up with lots of thermodynamic minima. We were ending up in something, because it's a kinetic process, something that is some other kinetic state. But here it turns out that once you look at through everything in whole composition and sequence space, you actually see that the most of the sequences code for thermodynamic ground states. So the AB sequence is a little bit misleading, giving you an idea that it's not only thermodynamic minima, but actually, if you look carefully, it is. Now, whether this persists with 3 colors, this is something that we are still exploring. With all the data that we have, we can do all kinds of classifications. We can try to look at these different geometries, try to understand how they look like. So I'm depicting some of them here below. We often end up with ones that have like a middle and the outside. Then we end up with something that looks polar. So it has one color on one side, another color on the other side. And we end up with shells. So there are rough categories of structures we can end up with just 2 colors. And we don't get other ones. So that's we think this is interesting. We are still trying to understand if we can actually extract from all the data that we have a more concrete conclusion. Then what we also have, we have an idea of how these sequences actually fold. The ones that have these longer stretches of blocks of color, they end up making chiral chains. With alternating, they usually make these compact tetrahedral structures. With 2, they end up making pentamer rings. So there are some rules that are emerging. I mean, we're not completely done yet, but this is what we are trying to understand. We're trying to find good order parameters that we can analyze to understand, to classify these things, because it's an enormous amount of data, but we are hopeful that we should be able to say something, because there are patterns, we can see it with our eye, the patterns are there. Just a matter of finding the right order parameter to classify them, to cluster them. Then what we also have are, as I said, kinetics, how things are explored. So we can look at, if you go directly from 1-dimensional chain to 3 dimensions, or you go via specific 2D state, how good you are you better or not? We do this in experiments and simulations. And it turns out, which is maybe not unexpected, if you go directly from 1D to 3D, you are faster, you have a better yield, which is not, at least for me, it was not completely clear, because if you enforce a certain 2D structure, you sort of prevent the system of exploring certain kinetic traps in the folding tree, because you're enforcing something that will for sure go downhill without any traps. But apparently, although the traps seem to be important, the 1D to 3D is still better. So this is also the information that we have. And so I finished because I'm also almost out of time. So there are some things we understand, many that we still don't. We have lots of data, but we are trying to do classification of the different systems, different structures. So why sequences select certain geometries and not others. We found that the folding modes of how things are folding, you see the two colors, they persist in 3D as well, like we had in 2D. Yeah, of course, this is not all me, this is a team of ladies and Angus that we've been doing this. So Maitana was my PhD student, so she's on the postdoc at Austria. Yasmin is, of course, Pierre at NYU. Natalie is my new PhD student at ESPCI. I would like to thank my funding, of course, for all that we've done, and you for your attention. I'm happy to take more questions. Yes.
You said that the structures that most readily assemble are also the deepest energy minimum structures.
Yeah.
So that would mean that you just started from the monomers, these would probably also assemble, but maybe much more slowly, right?
That's the point. So if you do, so if you see, you're absolutely right. So these are experiments and simulations on homopolymers. So we don't look at colors at all. We're just exploring the energy landscape of a system of a given size. So we know from calculations what we should expect. And so the experiments are done at the scale again of an hour, right? So something that's manageable in experiments. We're not waiting for days. If they do that, this downhill folding process where they're reducing the temperature within this hour, so they end up with situations exactly like this. So for example, here, so this is 7 particles. So the bars are results from experiments and simulations. And indeed, you can see that you can end up with something that's, so this is the thermodynamic minimum, this one with just a line mark, but you end up seeing most of the lag. So really, so to see this one, you have to wait really for a very, very, very long time. You don't see it easily. You more easily see everything else than that. But apparently, if you take sequences, it just goes there. But yeah, you're absolutely right. This is what happens. You have to wait for a very long time.
Really nice. A lot of questions. I'll try to ask one sort of open-ended one, and maybe you have to comment on it. I presume it's not relevant to experiments. I don't know if it's relevant for biology, but it sort of strikes me what's happening is that, you know, and it works very nicely in 2D, you have starting with like a linear subgraph of your triangular graph, and that is pretty easy to figure out how to sort of see local structure. Is there any reason to explore, or do you have any thoughts about looking at branch graphs?
Yeah.
Maybe this is more important in 3D than 2D.
Yeah.
Frustrating.
Yeah. So we looked at the branch stuff in the context of replication. Actually, so a different topic, but it's related to this because so in our replication system, so we are replicating from a surface of some structure. So we are transferring information, and then whether what you're replicating, if you're a polymer or a branched polymer, or something different, it depends a lot on how fast or how well you get to the replica state. So I think branch polymers are very important. Here we haven't explored them because the experiments turned to be so neat. So we were just looking at these ones. But the point is that in these folding trees, the way that we make them, what is missing the top row, I just showed a polymer. It's actually the huge tree which we have all the branch polymers as well that lead to there. So we have that information, we haven't explored.
Very cool.
Yes.
Yeah, absolutely fantastic. So what you could do is kinetic models on these split streams, where you answer the question, everything that you see at a certain level, you enumerate, and you go backwards and you see where there is a decision for one pathway ending up in one or the other. What we found, we did this for viruses and we find that if we have some species that are not really abundant, it's just circulate. Sometimes one or two splits, where it just favors material to flow along a dominant path, and you can characterize where this happens and when. And then if you want to design the system and then use this to your advantage, you can actually tweak, you know what you need to tweak to make it flow the other parts.
Yeah, no, so you're absolutely right. So this is the part that I haven't showed at all. But so with Eric van den Eyden, even with Maitane, we've been doing these games. So we have the folding trees, they are experimentally informed folding trees. So we have the rate information in some sense, and then we do the optimization thing. So how can I go here? So in non-equilibrium path oscillation, how can I go there? And then this particular state, what is the way to optimize the probability to get there? Because indeed what you said is correct. You sometimes just have one link, one step where everything breaks down exactly, and if you can force the system to go there, you are faster there, you are better. It's harder. Yeah, so we are looking into that as well. Yes.
Thank you.
Yes.
I understand that you use just random motion as the dynamics that lead you to the structure that you wanted to self-assemble. Would it be interesting to study the dynamics of these colloids in shear flows, for instance, where you might bias the yield rate?
Yeah, no, so that's an excellent question. There was a paper, I think last year, I think this was PNS, where they demonstrated, this was just computational completely, but they demonstrated how you can select certain structures in the shear flow by they were playing with different parameters of the flow. So that I think that's a very promising way to actually do stuff. Absolutely, we haven't done that, but I think that's a very promising way to go. Yeah.
It's like for your explanation method, can you like now update if you like have the ROMs and then let them self-assemble to see what structure they give? So just now using
Please, I did this.
No, I did it.
Yeah, okay, so I'm not showing you. I was sure I did it, but apparently not. Come on. Yeah, okay. So in simulation, what we are doing, we are trying to make quasicrystals, and so we are starting from rhombus, except that it's a binary system. So there are two types of rhombus, but they're made out of particles of two different sizes. And yeah, by tuning interactions, we can make lattice. Yeah, well, we've been playing with this only. We haven't done more than that.
We are enforcing the matching rules to ensure that it could potentially expand.
Yeah, so so this is sort of more dream. It's not exactly working 100%, but we are almost there. But indeed, we are enforcing the matching rules to interactions, and it's again this sequential protocol that actually allows you to gently go where you want to go.
So there's a full literature on how enforcing the matching rules and self-assembly and ultimately how it doesn't pan out, I guess.
Yeah.
So I guess I'd like to ask you about that. Are those black things defects, or are those part of the structure? You must have defects.
Oh, for sure. I mean, so this looks more like a random tiling quasicrystal, then it's not really ordered, and I'm not daring to show any crystallographic image of what this looks like.
Ultimately, that's kind of probably all you can.
Yeah, but no. So it's for sure they're defects. This is just random tiling. We are simply playing. So Michael Engel said something last week. He said actually it's super easy to make quasicrystals, and it's true in his system. So he had this tetrahedra, and they actually do form, you missed it. But it's recorded. You can find it online. So it's recorded. So it turned out to be super easy. That this is how he. So yeah, exactly. So they come together. They nicely align. They have experiments where they have these huge chunks of particles. It's absolutely amazing. So yeah. So we don't have that. What we were more interested in is how difficult it is actually to get it, right? Because how much do we really have to think really hard to get it in terms of designing interactions, or actually it's not that difficult compared to other things? So that was more of our motivation. But in any case, yeah. So we have the rhombuses that also work. But then the ones that this one is nice. So we have all kinds of networks. We're trying to fold into shells.
Oh yeah, that was my next step. It makes really case back in it.
Yeah, exactly. So at the moment it's still not working in simulations, but it's a technical issue because it has to work. I'm 100% sure. There is no other pathway of any. So there.
Certain angle that it can.
No, there is no angle. No angle. There is no. That's the whole point. There is no angle. What is only missing from this picture? So once you've made the two-dimensional structure, what is not, I cut it out. There is a small little particle that's floating around that likes to sit here in the middle, and it glues there. And once you lift everything in 3D, it just wraps up around it and nicely locks with all the interactions designed, and it just has to work. So the problem is that it's technical. I'm 99.9% sure. Yeah, well, we'll see. I mean, I like to be surprised, but there's no other option. That's the whole point. I mean, we constrained the landscape, the folding landscape so much that there is no other option. So it's technical. Yes.
Thank you. Can you just expand a little bit on in how far is the mimicking life?
Okay. So okay. So when I look at this picture, which is a picture of life, and there are all kinds of protein complexes that are there. There are lots of symmetries there. There are lots of repeating patterns that you see within the different machinery markets, right? And so there is some pattern there. I mean, there are for sure not all the possibilities, but there are some that are selected. They're better. They've been better for some reason, and others for not. So what we are trying to understand is that if we are trying to reach some the sport of emergent like phenomenon, in this case was it's not the emergent, but we were looking at the reliance on sensors, when we were looking at education, catalysis and other things. We're trying to see if there are some geometric rules that come up when we try to do this, right? So we are mimicking light because we are looking into emerging properties that we know from light, but along the way we are trying to understand, are there some geometric rules or pattern rules, you know, that merge that you don't see when you look at this, that are maybe crucial for making it functional.
As far as is life the limit of your experiment where you send n into infinity?
No, so I'm not going to define what life is. I'm not going to none of
but to put it differently, so as far as you've got a small number of molecules that are connected in sequence, so that will be one difference to what's happening in life. But then, of course, the other one is, so when you look at living matter, right? Typically, the temperature differences to go from one conformation to another are really quite small, whereas yours really seems to be largely an energetic rather than an entropy driven. Can you comment on?
Yeah. So again, most of the motivation was experiment with this was how it was done in experiment. So what I haven't showed yet before is that by looking for the different protocols, we can actually find protocols that are better at doing stuff than others. Meaning that if I take a bunch of chains of different lengths with one protocol, I will end up with a collection of things that can make tubules, assemble myself, and do some other things. There are some protocols that are more. Now, again, the simplest way to do these things is protocols, because this was the cleanest way to select secondary interaction. But now that we understand this, we want to do things at a fixed temperature, right? Because this is, we don't want any kind of variation. It's here more is needed, right? And so some things we can probably also realize with the fixed temperature, right? Playing quite a lot with the kinetics. And we have this cognition, we know what are the crucial states that if visited enable us to get somewhere where we want to go. So we can design these interactions as if we're targeting certain states, but then we'll get us there. So there is a way to optimize bond strength that will help us again remove for something the temperature protocols. But then what if you don't manage it, you won't manage for sure to remove it completely. It's suggesting it's giving different mechanisms. Here you need membranes or clock additions. Things have to be part of a process, right? Are they not part of anything yet, right? It's telling you that you need something else in there, some different mechanism, something different to be able actually to do the same thing on at the fixed temperature. So it's, for me, it's making me think about, okay, so now what's the next bigger different ingredient that I need to actually make this work? So I know how to do it in one limit. I want to do it in this limit, but I clearly have to add more stuff. And it's not trivial stuff. And you can only do so much interaction. Maybe actually solutions you have valence, you have bond angles. There are many different things that can help in this whole process of assembly, which we don't have here. And they're very big story. But for sure, clock addition, membrane, compartment, doing things, being something needed in part of the process, for sure they come into the story. So my hope is that this will come out at the end of the road. Does that answer the question?
Yes. When you stick together emulsion droplets with some adhesive energy, you might get a nice strong interaction. There will be a distortion of the spherical shape of the droplets, and there is also some capillary energy involved. Is this a contribution to the free energy that you consider in your modeling? You could have surface tension as another control parameter.
Mm-hmm. No, so Yasa together with Sasha exactly. They developed all this theory because they studied really this very, very carefully, because they were focusing on exactly this interface, how it looks like, what is the distortion of the interface. So here we have not looked at it whatsoever, but for sure there is an important because here, for example, valence was really not something that was very important. Whereas when you start considering valence, which you can, because you can control the number of DNAs and the emulsion droplets, so which means that if you control that, you can control how many bonds are involved in which interaction. Then these things matter. So there is a beautiful paper on that that's published. We haven't considered it here, but yeah, I think this is something to think about for sure in the future.
Thank you, Zorana.